Today we're talking about how to dissect genetics problems so that we can solve them efficiently. The success criteria is being able to determine genotypes and phenotypes in simple Mendelian problems. So this is actually pretty straightforward. You just need to pay close attention to what the question is asking for. We can do this in a 4 or 5-step process. First thing we want to do is identify the number and type of characteristics involved. Usually this means is it a monohybrid cross, is it a dihybrid cross with two traits, and if it is a dihybrid cross, does it involve epistatic genes at all? Will the two traits be inherited independently, or is one trait going to affect the other one? Second thing we're going to do is identify the traits that are available. This means figuring out what the alleles are. What are the traits? What are the alleles that go with those traits? Then we're going to identify what the parental generation is. This will depend on the information the question gives you, but we need to figure out what the genotypes provided are. And you always want to write these out because you want to be able to see what the gametes will be possible. Then we are going to use a Punnett square to determine what the F1 generation is. We can use that to figure out what the genotypic and phenotypic ratios are, and we have to bear in mind that if this is where we end, the final answer may be asking us for a ratio, a percentage, a fraction, or a probability. Now, in some questions, one generation will not be enough. We're going to have an F2 generation as well. So if the F2 generation is required, we're going to have to figure out what the parents in F1 are. Likely this means that everyone in F1 is going to be the same. And we're going to have to do a second cross of F1s using a Punnett square to figure out what their genotypes and phenotypes are.